Hi everyone, thank you all so much for joining us tonight. We'll be talking about the synthesis, in vitro, and preclinical in vivo evaluation of triacetate and acetonide analogs of Proscillaridin A. As Dr. No introduced us, my name is Shreya Samani. I'm currently a junior at Lindbergh High School.
And my name is Sini Menta, and I'm a senior at California High School.
So let's get started. Today we'll be talking about the introduction, which includes the advancements in oncology and HER-VK as a target. We'll then go through the synthesis of our analogs, including the high-throughput screening that first made them come to be, as well as the synthesis of our two novel compounds. We'll look at in vitro and in vivo breast cancer studies and our colon cancer evaluation, which we are currently working on. So, as you all know, cancer is the uncontrolled growth of cells, and there have currently been several advancements in the space, including but not limited to CAR T-cell therapy, chemotherapy, and more. But the question still bodes to be, what about untreatable cancers, and what drives some of the most aggressive cancers? Our collaborators who have worked with us on this project are interested in a protein called HER-VK. HER-VK stands for Human Endogenous Retrovirus K. And it was a virus that was actually uptaken by humans several thousands of years ago and was integrated into their genome. Scientists within the last few decades have noticed that these proteins are actually visible on cancerous cells and can be seen budding off. Due to that phenomena, we have begun our search for an inhibitor of this protein, as no known one has currently been discovered. This started off with a high-throughput screening, and high-throughput screenings are incredibly common in the medicinal chemistry space, as it allows a company to take several thousands of possible compounds and narrow them down into a few target ones. Our high-throughput screen in specific involves several different groups, such as natural products, steroidals, immunomodulators, and more. We screened over 100 of compounds in our chemical inventory against HER-VK transduced cells with our collaborators. Now, these results actually showed that we had one compound, which was a hit, and that was digoxin. Digoxin is a cardiac glycoside, and it is a sodium-potassium pump inhibitor. So it actually hasn't been discovered as an anti-cancer molecule. This compound has both a steroid and a sugar, and once we discovered this one, we then tried to further expand our library.
So like Shreya said, after we found digoxin as our hit compound, we decided to further look into other compounds that pose both that sugar and steroid group. We found both. We first started with finding digitoxin, very similar to digoxin, but then after that, we looked into Proscillaridin A. And due to Proscillaridin A's efficacy and known potency in comparison to digoxin and digitoxin, we decided to synthesize two analogs, proscillaridin triacetate and acetonide. Both we were able to yield over 75%. With that in hand, we had 5 compounds that we wanted to evaluate further in various biological assays. We first initiated the MTT assay, which is a cytotoxicity readout. Due to our collaborators' interest in the breast cancer space, we evaluated our compounds on various breast cancer cell lines. Some of them include the MDA-MB231P, MDA-MB231K, which are differentiated because the K is transduced with the HER-VK protein. We also did other cell lines such as 468 and the MCF7K and MCF7. With all these results, we were able to see great potency in comparatively within all our compounds, and due to that, we decided to further our evaluation in an in vivo study. Right before the in vivo study, we tested our compounds on 4T1K cells, which are a metastatic mouse breast cancer cell line. And due to great potency within that, we were able to move on to the in vivo study.
So as Cindy mentioned, as our compounds had great promise and 3 of them had already been tested in vivo trials, we then moved on to the in vivo study of one. Now this was performed with 36 different biopsy female mice, and of course it was in collaboration with our collaborators from Sunny Bay Biotech. Now these mice were injected with the 4T1K, which as she mentioned is a metastatic breast cancer tumor, intraperitoneally. And after 3 days of tumor growth, they were subjected to different drug injections on Monday, Wednesday, and Friday. Now, during this time period, it's not just the injections that were happening, but also all the housekeeping that goes on with an in vivo trial. This includes the weight measurements of the mice, their tumor mass measurements, and the cage maintenance. Now, this trial lasts for over 50 days, and as you can see here, this is some preliminary data, well, not preliminary, but first disclosed data, which shows the progression of the tumor size as the days went on. The arrows indicate drug dosing, and we unfortunately had to stop giving our drugs due to different off-target toxicity effects, which we'll talk about in the next slide. But as can be seen here, we see that the control actually showed a complete growth of tumor as they had not been subjected to any drug, and that was simply just a placeholder in a placebo. The addition of our compounds showed decreased tumor size, with Proscillaridin A and its analogs having some of the lowest. This can be better seen in these different bar graphs, which show the mice survival rate and relative tumor volumes. Unfortunately, as I mentioned before, due to off-target toxicity, which means that the drug can enter the bloodstream and travel to different parts of the mouse's body and have different toxic effects not related to what we wanted, we did see that the mice subjected to our Pros-A analogs had a lower survival rate. However, this is in tandem with having decreased tumor volumes as compared to the control. We also saw that our two analogs had the best mitigation of the tumor volume, with Proscillaridin acetonide being the best, as we also observed in our in vitro assays. Now, this concluded our in vivo study, and we actually wanted to shift gears into a different type and explore how these compounds would behave in a colon cancer cell line. Now, this has just been some data that we've been currently working on in the past 2 months, and we started off once again with the very general and standard MTT cytotoxicity assay. This assay measures the viability of cells in a sample over the current time period in which we did 24, 48, and 72 hours of administration with our drug. Once again, as you can see here, there's a clear trend that shows that oftentimes Proscillaridin A and acetonide will act the best as compared to our panel of compounds. And this also shows the greatest toxicity is at 72 hours, which makes sense since the cells have been drugged with the compounds for longer. We performed this assay on 3 different colon cancer cell lines, HC2116, HC29, CT26, and HepG2, which is a liver carcinomic cell line. We did it on a liver cell line just because we wanted to see if anything could be studied based on the metabolism of these compounds in a liver cell line. So after we performed this very simple MTT assay, we then turned to the LDH, or lactate dehydrogenase assay. This assay measures the bursting or the necrosis of cells in a sample. LDH is an enzyme that will convert pyruvate into NADH, which can actually be measured spectrophotometrically with a colorimetric substrate. And this assay actually showed very low lysis or very low LDH release within our compounds, which also tells us that these compounds may not or probably do not act through actual cell lysis and bursting. Now, we also wanted to kind of probe the mechanism of these compounds without further elucidating it, and so we looked into different reporter cell assays. One such assay that we looked at involved the HEC293 cell line, and we wanted to look at its activity with NF-kappa B. To do this, we performed a SEEP assay, which is a secreted alkaline phosphatase assay, with a stable transfected reporter cell line, in this case, HEC293s. The addition of TNF-alpha, which is a tumor necrosis factor, to these cell lines alongside the administration of our compounds resulted in the activation of the NF-kappa B transcription factor, which then is followed by translocation and the binding of NF-kappa B to the promoter. The promoter's activation can actually be measured with the colorimetric substrate, and so we performed this assay to get a hint at what the mechanism could be. We wanted to do it on both NF-kappa B and CD40, and as you can see on the left, they both have very similar pathways overlapping. This assay showed that all our compounds actually shut down TNF-alpha-induced NF-kappa B signaling and CD40L-induced CD40 signaling. As one of our last reporter cell assays, we tried to do one with the STAT1 inhibition pathway. STAT1 generally acts as a tumor suppressor, And here we can see the potent inhibition of this with all of our compounds as well.
As our final biological assay that we conducted on all our compounds, we decided to do flow cytometry analysis. Briefly, flow cytometry is an assay that is able to analyze the individual cells that are passed through a laser beam. We're able to identify the light scatter and also the fluorescence emission that is measured quantitatively. We're also able to see various cell qualities such as cell size, cell cycle phase, appearance, activation of proteins, and also apoptotic mechanism. The first one that we conducted was apoptosis flow cytometry. Like I said, apoptosis is a mechanism we're able to kind of conduct through this. And apoptosis, also known as programmed cell death, is something we're able to track through the different stages. Here we can see on the left various graphs that indicate the 4 stages that apoptosis is present in our cells. As we can see, most of our cells are found in the left bottom quadrant, which is the viable percent. As we go from left to right and up to the top, it goes from early apoptotic, and finally necrosis. However, we can see that most of our compounds, in compared to the control, don't really act through this apoptotic mechanism. This was done on the HCT116 cells. However, we were able to replicate this also on the HT29 cells at a higher concentration, but did not see much differentiation. We did see very clean graphs showing that our assay worked very well, however, very comparable to the control. The next flow cytometry assay we ran was cell cycle flow cytometry. This in particular is able to track the phase of the cell cycle that the cells are in. As we know, the cells can be in G1 or G0, S, and M or G2, which is the main mitosis phase. As we can see on the right, this is what a graph of the cell cycle flow will look like. The peak on the left is the G1 or G0 phase, the middle will indicate S phase, and finally on the right is the G2M phase. Here we are able to quantify if any of our compounds are being inhibited at a specific checkpoint or a specific phase. So starting from the left, we started at the CT26 cell line. We did all of these at 100 nanomolar. However, we weren't able to see much differentiation from the control once again. This can lead us to conclude that our compounds do not really act through cell cycle inhibition. And due to that, we decided to see if we can evaluate it further in protein marker flow cytometry. So first one we did on the left is the CD117 assay. This is a CD117, also known as c-KIT, is a tyrosine kinase inhibitor. And CD117 is usually overexpressed in cancer cell lines. So an ideal result of our compounds would be the inhibition of the cancer cells. As we can see, it is normalized to the DMSO control at 100%, and all of our compounds do show decrease of proliferation. Furthering that, we did CD133, which is also another cell surface protein that is overexpressed in cancer cell lines. However, we weren't able to see much differentiation from the control with this experiment, and we hope to further replicate this to see if we can evaluate it further at higher concentrations. Finally, we did caspase-3. However, caspase-3 is actually an enzyme that is an enzyme in apoptosis. This does really build off our previous apoptosis results, as we weren't able to conclude much from that. So caspase-3 is actually activated in response to cell damage. So our compounds ideally would lead to higher caspase-3 activation. So as we can see, most of our compounds do that besides digitoxin, and we hope to also evaluate this further at a higher concentration to see if these results are replicated. In conclusion, we were able to synthesize 2 novel glycan Proscillaridin A analogs and evaluate them in various biological assays. As we can see, we weren't able to find much apoptotic or apoptotic inhibition. However, we did know that there was great anti-proliferative activity. We did not see any activity in the LDH assay and little to no activity in the pro-apoptotic and cell cycle inhibition. Finally, we did see CD117 cell surface receptor inhibition of digoxin, Pros-A, and the Proscillaridin acetonide that we do find significant. And finally, we found that the reporter assays do indicate a shutdown of TNF-alpha-induced NF-kappa B signaling and the CD40-induced CD40 signaling as well. We'd like to take a moment to acknowledge our collaborators and also Dr. Edward New for helping us along with this project. And of course, thank you to our lab group members. Thank you.